I'm going to cover Unit 1, Part 2B, and begin with metabolic pathways concerning energy. So again, this is all completely review from previous science courses, but I will add a little bit of information since I know this is at times a tough topic for students. So first we have cellular respiration, which has both an aerobic and an anaerobic pathway. And then we also have photosynthesis. There is one aspect of these two equations that students don't always remember, so I want to start by pointing that out. So for cellular respiration, essentially your reactants or beginning products are glucose and oxygen, and then your end products are carbon dioxide and water. So this is pretty much the complete breakdown of glucose to provide energy for living beings. And if you take a look between the two equations, what you're essentially seeing is the end products of cellular respiration are actually the reactants or the start products, and therefore cellular respiration is beginning with oxygen and glucose, whereas photosynthesis takes CO2 from the air as well as water, and your end products are glucose as well as oxygen. So let's first mention aerobic metabolism. I'm going to put a couple of notes on the bottom here in hopes that you remember this. And if you don't, definitely look at a video on cellular respiration and aerobic respiration to ensure that you are comfortable with the topics. So with cellular respiration, there's essentially three components. There is a fourth one, which is like a transition in the middle, but we're going to stick to the three main ones. First, we have glycolysis, and I'm going to focus on how many ATP are made. At the end of glycolysis, 2 ATP are produced. And again, we are talking about aerobic respiration. The second step is the Krebs cycle. For the Krebs cycle, again, at the end of it, you have 2 ATP that are produced. The third one is known as the electron transport chain or system. That's why you see an S over here for system. And here, we're going to say there are 30 plus ATP produced. With the first step of glycolysis, another important aspect we have to think about is pyruvate. With this particular process, there is 36 ATP that is produced. It is very efficient because ATP is the energy yielding molecule. And again, this is aerobic with oxygen involved. You will need to know that you'll see on the next slide where within the bacteria each of these three steps is taking place. So now if you look at the picture above, again, you have glucose, which yields 2 ATP and pyruvate. You have Krebs cycle, which yields 2 ATP that plugs into the electron transport chain or system, where you end up with a total of 36 ATP. So glycolysis, which is the first step, denoted by the G, occurs outside of the bacteria. Then the end products of not only pyruvate, but these 2 ATP go inside of the bacteria, where the second step takes place of Krebs cycle. So this one is inside of bacteria. And then finally, those 2 ATP move into the electron transport chain, which occurs within the membrane of the bacteria. This is, again, aerobic respiration where oxygen is present. Notice that you see O2 written throughout this entire picture of the bacteria. There's a statement that you need to be able to answer to ensure that you understand. And that statement is that the fate of pyruvate is dependent upon the availability of oxygen. What that means is, after the first step of glycolysis, what happened to the pyruvate? It went inside of the bacteria, which then fed into the Krebs cycle and fed into electron transport chain. This happened because there's oxygen present everywhere, and this is an aerobic respiration example. So again, here is aerobic, but now we're going to talk about anaerobic. And anaerobic means that there is no oxygen. The yields are very different. So what happens with anaerobic is you have your first step of glycolysis, like we discussed in the beginning, which yields your 2 ATP and pyruvate. Because this is anaerobic, that means there is no oxygen, and therefore there is also no Krebs cycle. That doesn't happen. In anaerobic, here are the steps. There is glycolysis, which is again your pyruvate and 2 ATP. There is no oxygen, and therefore there is no Krebs cycle. Instead, the second step is fermentation, which also yields 2 ATP. So at the end of anaerobic respiration, you are looking at a total of 4 ATP. Therefore, it is not as effective or efficient. So when you think back to the statement I wrote out before, the fate of pyruvate depends on the availability of oxygen. If there's oxygen present, pyruvate feeds into first the Krebs cycle, which yields 2 ATP, and then the electron transport chain, and in aerobic respiration, your final yield of ATP is 36. In anaerobic respiration, the fate of pyruvate is dependent on oxygen because there is no oxygen available. So the pyruvate made from glycolysis falls into fermentation, and then fermentation yields 2 ATP, so anaerobic yields a total of 4 ATP. Not as effective or efficient. There are 2 types of fermentation. There is the one related to alcohol fermentation. These are, think of food and wine and different products, as you can see here. And the other one is lactate or lactic acid based fermentation, which is also anaerobic, or again lacking of oxygen. So to show you what's happening, this picture depicts anaerobic, and make sure you go back and compare this to aerobic. You have glycolysis occurring on the outside of the cell, so same as before, this is happening on the outside. Is there oxygen present? No, so we're going to cross off all of the oxygen. It is not present. So therefore, the pyruvate is made, and it does not go into Krebs cycle. And if there's no Krebs cycle, there's also no electron transport chain. Instead, the pyruvate and the 2 ATP from glycolysis is fed into fermentation. So again, as a review, for anaerobic respiration, the first step is glycolysis, where you end up with pyruvate and 2 ATP. Remember the statement, the fate of pyruvate is dependent upon the availability of oxygen. Well, this is anaerobic, and therefore there's no oxygen, so there's no Krebs cycle, there's no electron transport chain. The end products of glycolysis feed into fermentation, which again yields 2 ATP. So for anaerobic respiration, the total ATP count is 4 ATP. It is not as efficient in producing energy-yielding molecules as aerobic respiration is, which produced 36 ATP. So again, you go from anaerobic to fermentation. Why? Because there's no oxygen, which means there's no Krebs cycle and there's no electron transport chain. Glycolysis yields 2 ATP and fermentation yields 2 ATP, and anaerobic respiration has a total yield of 4 ATP. So now a mini little look at photosynthesis. Again, cellular respiration, the beginning products were were glucose and oxygen, which then yielded the end products of carbon dioxide and water. Whereas photosynthesis, this arrow is simply reversed, and the beginning products are carbon dioxide and water, and the end products are the food, which is the glucose and the oxygen. Remember, in bacteria, we're not talking about things like chloroplasts because these are not plants. We are talking purely about the cell wall. As we mentioned in the first part of Unit 1, PowerPoint 1, there are two ways that bacteria yield their nutrients. One of them, they absorb nutrients, and the second one is by photosynthesizing. This is the definition and the equation for photosynthesis. So we're going to switch gears completely. I would really ask that you look up some type of simple video. If you really don't remember, notice I didn't bring up oxygen electron acceptors, inorganic organic compounds, NADP, FADH. I brought up none of those components. I just need you to know aerobic versus anaerobic, where in the bacteria it occurs, which is more efficient, meaning which one yields more ATP, what are the actual steps, and to truly understand the statement that the fate of pyruvate depends on the availability of oxygen. If it's available, we go aerobic. If there is no oxygen available, the pathway is anaerobic, which is fermentation and not as efficient as aerobic. So what we're switching to now is a sheerly microbiology thing and completely different. There is a proper way to name organisms, and not only how to name them, but how to actually properly write their names. So on every single one of your exams and the case studies and the homework assignments, you will be docked points if you don't write or put the name correctly. This is known as binomial nomenclature. Nomenclature might make you think of Chemistry 130. Bi means two, so it is a two-word naming system for bacteria. So this example here is Escherichia coli, but first that's basically E. coli. I'm going to use a completely different organism on the right here and write it out for you to make it a little simpler to explain. Everyone has heard of Staph aureus. The actual name is Staphylococcus aureus. When you write the name of a bacteria, you have to do one of two things. You either underline it, or if you are typing, you can italicize. And you cannot italicize written because then it just looks like you're writing sloppy. So there is the group of organism, which is Staphylococcus, and then aureus is the specific type of Staphylococcus microbe that we are discussing. So again, you have the first and the second name, and then you must underline it. The first letter of the first part of the name is always capital. The second letter, or the first letter of the second name, is always lowercase. Another way to write this is you basically shorten the first word. In other words, you can have S period and then aureus, but remember the S has to be capital, and the first letter of the second name is short, and then again you underline or italicize if you happen to be typing. So if we have Proteus vulgaris, I want to again point out the P is capital, the V is lowercase, and then again I need to underline it. If you want to write the shortened name, it is a capital P, and then vulgaris, and what do? You underline it. So this is binomial nomenclature and the proper way to write the name of an organism or bacteria. So if you take a look at E. coli written out here, Escherichia coli is crossed off right here because it's not underlined and it's not italicized, so that is not correct. You do have the capital E and the lowercase c. So now if you look at the second one, this is in fact correct. The first letter of the first word is capital, the first letter of the second word is lowercase, and notice it's italicized. The third one is also correct because it is in fact underlined. The fourth one is correct because it is italicized, the E is capital, the C is small, and 0157H7 is the pathogenic strain of E. coli. This is actually called the variance or variant, and it is the strain number. Most bacteria we work with will not have that. And when you add the variant at the end, which is a grouping of letters and numbers, whether or not you italicize or underline it does not matter. This is only relevant to what you actually do with the organism, the words, the name, the binomial nomenclature. So one more example up top on the right. There's an organism known as Bacillus cereus. Notice the B is capital, the C is lowercase, and then I need to underline it. If I want to do this in its shortened form, it's a capital B, and then a lowercase cereus. I absolutely need you on your exams every single time and all of your homework moving forward to either italicize or underline. Make sure in the first letter of the first word is capital, the first letter of the second word is lowercase. After one or two assignments where you lose 1 or 2 points, you'll remember, but this is incredibly important because it is in fact the proper way to name bacteria. Outside of binomial nomenclature, which is the names, we have to group bacteria based on some type of phenotype. It is very difficult to look at bacteria growing in a tube and know exactly what it is. If you take a look at the picture on the right, these are two different organisms, whereas both of them essentially look blue. The control and the E. coli are two different organisms, and they essentially look green. If you take a look at these tubes on the end, this is a biochemical test that you'll run across when you get a little further along in lab. Those tubes, upside down tubes, are called Durham tubes, and they're collecting gas. But if you just simply look at the color, I would say these three are yellow and these two are red. So there's no way to phenotypically just take a look at bacteria and know exactly what they are. There's a very specific way that we classify bacteria, and that is using the 16S rRNA. rRNA is ribosomal RNA. If you think about when you learn transcription in your BIO205 high school biology or BIO181 course, it was usually referred to as a hamburger bun, where you have the smaller top part of the bun and then the larger top of the bun, and then the mRNA is growing as a tail in between as you are doing, or the process of transcription is occurring. So the way that we classify bacteria has to be more genotypically and not at all phenotypically. Why? Because if you think back to the previous slide, the colors are so similar and you cannot identify it by quickly taking a look at it. So what is used to identify bacteria? 16S rRNA. Size is another factor when it comes to bacteria and classifying it. In previous biology courses you've taken, it's always been the compound light microscope, which is on the bottom here, that is looked at. You did not need to stain it. You can simply look. There was the colored thread. There was the Volvox. There were little pieces of, say, potato or onion so that you can actually see the cell wall and the different components. But I need for you to be comfortable understanding the size. So bacteria are measured in micrometers. This is like a cursive letter U. And you notice micrometers fall right here. This is pretty much their size range. You would in fact use a compound light microscope to look at them. Things like fungi and protozoa are much larger, but we'll look at fungi. These can be looked at the unaided eye. What that means is you can see mold and yeast growing without actually using a microscope to see them. Then on the other hand, you have viruses. Viruses are incredibly small, and they are measured in nanometers. You cannot use a light microscope for viruses. In fact, you need to use an electron microscope. So in summary on the side here, I will tell you that bacteria are significantly larger than viruses, and they are measured in micrometers, whereas viruses are much smaller, and they are measured in nanometers with the use of an electron microscope. This is a picture to give you a size understanding of how small micrometers are for bacterial measurement. This is a pin or a head of a pin, so if you buy a men's shirt, usually in the collar, brand new when it's folded, it'll have a couple pins, or if you go to a seamstress, this first picture is the head of a pin, and this is at 100 micrometers, and then 20, and then here the last one is magnified 14,000 times. So on this one head of the pin, look at all of this bacteria that would absolutely fit on it. So bacteria are very small. It measured in micrometers, and remember, viruses are even smaller, measured in nanometers. Lastly, I want to point out that there are certain terms that I would like for you to be familiar with. They are listed for you on page 3 of your outline, and I did mention this in the lecture outline as well, that you should know them. A couple of medical microbiology terms. When it comes to bacteria, less than 1% are pathogenic, which means infectious. Most bacteria are our friends. So pathogenic is the level of is it or not level, but is it or is it not infectious? If it's pathogenic, it's infectious. If it's non-pathogenic, it's not infectious. This does work both ways because we have E. coli that live as normal flora in our GI tract. Those are non-pathogenic. Then there's the H1057, the one that we had discussed before when we were doing binomial nomenclature. That is the pathogenic one that would then cause GI issues. Virulence is another term that's very important, and this is the severity of a disease. Why does that matter? Because if the virulence level is low, that means the disease is not so severe and the symptoms aren't so harsh. Please keep in mind that except for viruses, more tend to be pathogenic. Bacteria, as a large overall whole, are non-pathogenic, and many of them live as normal flora in our bodies. So a couple other terms to mention. Disease. Most people go, oh, I know that word, but you really need to understand what it actually means. And the definition for disease is basically away from the norm. Whenever you're ill, you are essentially diseased, which means some physiological function is away from what is normal in your body. So disease-causing microbes are pathogenic ones that push your body away from the norm when you become infected with them. Then the virulence is how severe or not severe they are. So the picture on the left are the pox from someone who has chicken pox, and the pictures on the right are in fact syphilis. The top picture is an open sore related to the later stage of syphilis on the shaft of a penis. So could you say that in both cases the patients on the left as well as the right are diseased? Absolutely, because chickenpox is away from the norm and syphilis is away from the norm.